Well, welcome to this seminar of the cycle of scientific seminars of the Roca Solano Institute. As you know, we have with all of us to Javier Buceta, which I acknowledge your acceptance to participate in our cycle. Thank you very much, Javier.
Mm-hmm.
So as usually, I am going to say a few words about his scientific career. Javier graduated at the University Complutense of Madrid in theoretical physics and also in computer analyst. After graduating, he went to, I talked to him about this because it's a very curious stay, in Hungary, in the Institute for Nuclear Research. And this is before starting his PhD. He made his this PhD on fluctuations in especially extended systems. And the focus of his research was pattern formation, molecular motors, and symmetry breaking phenomena. During this period of time, he made some stays in Italy and Belgium. Then he moved to his first postdoc at the University of California, San Diego, at the Department of Chemistry and Biochemistry, and in the Institute for Nonlinear Science, where he joined the group of Professor Lindenberg. This was his first postdoc. After that, he moved to the La Jolla, is this correct? To La Jolla Institute, in his second postdoc, where he conducted research on pattern formation and left-right symmetry breaking in embryo development. After that, he came to Spain, in particular to Barcelona, as a Ramón y Cajal researcher. And then, after a positive I3 evaluation, he was tenured at the end of 2009. Later in 2014, he accepted an offer from the Lehigh, is this correct, the pronunciation? Lehigh University in Pennsylvania, as an associate professor in the bioengineering program and the chemical and biomolecular engineering department in the area of systems biology. In fact, I think that the main discipline he works is system biology in general terms. After this, he moved to the Institute for Integrative Systems Biology, which is a mixed center between CSIC and the University of Valencia, which is the place he is working currently. And now he is making his research using many different approaches, from theoretical approaches, computational, and experimental ones. He focused his interest in problems concerning developmental dynamics, problems in microbiology, such as growth biomechanics, quorum sensing, and so on. And in particular, he pays special attention to stochasticity and the feedback between signaling and biomechanics. Well, he's very active also as participating in many different networks. Probably he's going to talk about this at the end of the of his talk. And I don't want to drop more time. And now, please, Javier, it's your turn. Thank you very much.
Thank you. So can you hear me well? So thank you very much for the invitation to the institute, for the opportunity to be here, and for this very nice and long introduction. So let me first say something in general about, I'm going to try to be as not deep into the details. I'm going to be in the institute for a couple of more days, working with my very good friend Pablo. So if you have any questions about details or things like that, just come to me, drop me a line, and I will be pleased to talk to you. The thing that I'm going to talk today is something about the structure of epithelial monolayers. In particular, as you can see here, with this Roman arch, something that we became interested is about how cells actually pack together when you have to try to make a structure that has some curvature. And I will explain why this is actually important. Okay? That being said, I'm going to start saying also that it's an incredible pleasure for me to be here in this particular institute, because it brings me back to the years when I was a PhD student. So many, many years ago, when I was starting my PhD, I used to come here because, and this is for the younger audience, believe it or not, internet was not always there. So it was at the infancy. So if you want to actually to know what was being published and these kind of things, you have to come to the libraries, look at the journal indices, make photocopies and these kind of things. And I spent a lot of time in this building in the library doing this kind of things. So it's actually a big pleasure for me to be here. There is another reason why I am very happy to be here today, is that it's actually the very first time in my life that I'm giving a talk in my birthday. So today is my birthday. So at the end of the talk, if you want to sing me something, I will be very pleased also to do that. Anyway, if you want to find out more about all the other things that we are actually doing, you can just visit the webpage of my group, and you will find the kind of things that we are doing. In general terms, we are dealing with three main problems at different lengths and temporal scales. One of the things that we are actually working is at a single cell level, working in microbiology problems. So trying to understand how mechanics and signaling coordinate with each other, the role of noise in these processes, and these kind of things. At a different level, something that we are actually working is at the collective cellular behavior, in particular in developmental biology problems. And then at a complete different length and temporal scale, we are working in the dynamics of full organisms, in particular oriented to understanding epidemiology problems. This is not something that I jump in because it's fancy, as you can see now, unfortunately, because of COVID. I started that line many, many years ago. And actually, I'm not working in COVID. I'm working in Ebola. So it's a different issue. Okay, so today, actually, I'm going to just tell you something about the developmental biology problems that we are actually working with. But as I said, I'm going to be here in the institute for a couple of days. So if you are interested in any of these things, just let me know. Okay. So in particular, or in general, something that we want to understand is shape. How shape is actually achieved in collective cell behavior. How cells are able to coordinate together in order to make a particular task that can be to create a form. Okay? And that actually involves, it's a problem that involves many different issues. So one of the things is gene regulation, how cells communicate with each other, how the internal signaling of cells is actually coordinated, these kind of things, and how this single or nearest communication between cells can actually achieve a coordination between them in order to coordinate other properties that are actually important for creating a shape, like how cells are actually elongating, and how that elongation at the single cell level actually can be translated into elongation or shape regulation at the collective or tissue level, how cells are actually dividing. That implies also mechanical problems. So this is the general overview about the kind of things that we are actually working with. One specific example about these kind of things is the formation of your hands. So this is not really your hand, but this is how your hand began when you were an embryo. So this is the so-called the Limba, and in the Limba, the size of this is around 1 millimeter square, more or less. And as you can see, there is a particular pattern of gene expression that actually continues. This pattern of gene expression is going to determine, or is going to tell cells that this cell is going to become part of the finger, or this cell has to die. And that is why actually at the end you end with your hand. So in this case, there is this problem about the gene regulation and gene expression that is creating a pattern. And at the same time is the mechanical coordination between all these cells in order to produce, in this case, a particular elongation in one direction. Okay? So it's a particular directionality in the process also involved. For doing these kind of things, we work with computer simulations. In particular, this is one simulation about this process where you have cells that are expressing some in silico genes that are able to develop a pattern, and then you have the growth of the cells, the cleavage of the cells, and a particular directionality that is actually achieved. Something that I'm going to discuss here today is that this particular problem has a specific conundrum that is very interesting. The point is that regardless that you can see that there is a particular directionality, something that is well known at the level of developmental biology is that when you look at the specific elongation of the individual cells, cells are actually elongating in the perpendicular direction of the elongation of the tissue. So it's actually a tricky problem because you have to understand how a tissue can grow in one direction, but actually the cells are growing in the perpendicular direction. If you want, I can come another day in order to tell you about how that mystery is actually resolved. Okay. In this particular topic, or in general in these topics, we want to understand also these problems from the point of view of if you were dealing with a material. Okay? So we can characterize these processes trying to understand the mechanics or the processes that are important. For instance, in this map here, by the way, the green square is how the tissue began in terms of size. The other square is how the tissue ended. So you can see actually that there is this directionality in the process. And here what we are actually in this density map is we are actually plotting where cell divisions happen. So when you are actually adding or having more cell divisions. So an additive process, if you want, from the point of view of materials. The other things that we want to understand is that how tissue actually regulates their plasticity. In this map over here, what you have is something that I will mention later, that is how cells can actually change their neighbors, intercalation processes. Okay? So how they actually move around, and that is also a particular, in this case, very important in order to achieve this shape regulation. Okay? For doing that, we are actually developing tools. This is one particular tool that is able to deal with these simulations about how regulate forces or signaling, and how actually this package, if you want to do a simulation of this kind of developmental processes, can be done. Okay. Now, I didn't mention this before. Probably this should be one of the first slides, but I decided to move it up to this point, is that normally I'm going to talk about epithelial cells. So an epithelial cells are kind of the building blocks of all, or not all, but most of the organs. Not only your skin, that is the idea that normally comes to mind when talking about epithelia. Epithelia are the building blocks from the inside to the outside of our bodies. So it's the kind of cells that we work with in order to understand these processes. Something that is particularly important about the epithelial cells is that they are actually packed tightly with one another. Okay, so it's like in the simulation that you saw. Normally, you don't see holes between cells and these kind of things. Okay. And in this context, probably Luisma that is watching this seminar, he told me online, you will know who is Luisma later on. I'm using here Lego. He's going to hate me by that because he's a Tente freak. But in any case, I'm using Lego here. The idea here is that if you want to pack together all these cells in order to build, like in this case, a particular organism, something that is very important is actually to understand how, as I said at the beginning, these cells pack together or the shape that they actually have in order to achieve one particular task. That can be elongation, or it can be actually building something that, as I will tell you now, can be actually in 3D. That is the problem that I'm going to focus. Okay. So I'm going to focus on 3D from now on because it's actually a problem that has been not really disregarded, but not really paying attention for a lot of time in developmental biology, mostly because the technical problems that we had in order to understand the structure of the cells in 3D. Okay. So how they pack together on 3D and all these kind of things were actually something not very clear for many years because of some limitations in the microscopic fields. And now that we can actually watch more in detail cells in 3D, we can actually try to understand better all these processes that in the previous movies that I showed you was in 2D. I want to explain you why. But actually they have a relevance, of course, in 3D, because all this structure has a 3D structure, architecture, and in particular, they actually go through bending and folding, and I love this sentence that all developmental biology know, that is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. So gastrulation is this process where in the embryo there is disinvagination here. This is going to distinguish mesoderm, endoderm. If this goes wrong, you wouldn't be here. I wouldn't be here. So it's actually very important. And in this particular process, in gastrulation, as you can see, folding and bending of cells is actually something very important. So it's not only here, but in many processes where bending or shaping tissues like that is particularly relevant. So when you typically look into books about, so how are these shapes, these epithelial cells? This is normally the image that you will find. You will see that it's kind of a prism-like. You will see that there is a lot going on in one of the surfaces of those cells. That a lot of going on means that most of the mechanical interactions between these different cells are actually concentrated in one part of the cell, that is the so-called the apical part. So these cells are actually polarized mechanically, if you want. And you will see something like this. And the reason why in the previous movie that I showed you about simulations, we many times focus on 2D is precisely because since all the polarized mechanical properties are actually on one side of the cell, the rest of it is actually disregarded and we consider like a fluffy thing that is hanging over there. But of course, if you want to understand a 3D, you cannot do that anymore. Okay. So what happens with those cells when there is folding or bending or these kind of things? When you have a flat epithelial, like in this case, people has rendered the cells like these prisms that I just mentioned. Okay, and how those cells are actually were thought to behave when you have bending or folding is like, well, the shape probably will be something like this. This is in math, is called a frustrum. This is a prism. Okay, and as I said, normally we assume that because normally what is imaged in microscopy is the apical part or the basal part of the cells, but not really the 3D structure. Okay. So this is something that actually we question, is that the case? It's actually really that is happening to the shape of the cells when you have bending or folding. And for doing that, the simplest model that you can actually think of is like a cylinder, okay? And it's simple enough because there is only curvature in one direction, not in another direction, not in the two directions, okay? And there is this computational, there is a lot of information here that I'm going to ask you to remember in a sec, okay? So in this particular cylinder, you can actually populate one of the surfaces, the inner surface, that is in this case the apical one for the cells, where more of the mechanical interactions are going to be concentrated. I'm not going to tell you the details unless you ask me at the end how you did it, but it's actually using what is called a Voronoi tessellation, okay? For this, you can determine what is the planar shape of the cells. Then you can project the centroids of those cells to the other surface, in this case the basal one, okay? And then in each of the intermediate values of this radius, you can actually reconstruct this Voronoi tessellation in order to render the 3D shape of the cells, okay? Now, something that came out from this simulation was this particular issue, that if in this particular group of 4 cells, you can actually see that here the blue and the yellow are neighbors. They are touching each other, but the green and the red are not. But then, after doing all these projections, we saw that in the other surface, actually the blue and the yellow were not neighbors anymore, and the green and the red were neighbors. So now, if you think about it, this kind of behavior is absolutely incompatible with this image of a prism or a frustrum. Because if you have a prism and you have a cell, remember there is no holes between cells. They are always trying to pack together tightly. You always maintain your neighbors, okay? From one surface to the other. But this is going to, I don't think that this can be seen in YouTube, but I have these 3D printed cells that actually, they are just reproducing what I just told you. So here you have the green and the red, that in this surface are neighbors, but here in this other surface they are not. And the opposite happens with the blue and the yellow, okay? Now the thing is that what is the shape of these cells such that they're able to achieve this neighbor interchange without leaving any holes or gaps between them, okay? So if you want to play a little bit with it, I'm going to pass you this around. Okay. So there was this very interesting problem that we need to solve. And there are a couple of quantities that I want you to remember here because they are going to be important later on. What is something or a variable that is called the surface ratio, okay? That is nothing but a dimensional radius in this cylinder, okay? This is actually a measure somehow of the curvature also and the thickness of this 3D tissue, okay? And the other one is the cellular aspect ratio. This is actually is a cousin, but at the single cell level of this guy, okay? So this is a measure of how curvature changes at the collective level, if you want, at the level of the full tissue. This is something that is telling you information about how that curvature is achieved at the single cell level. With these cellular aspect ratios, both of them can be related. What I mean is that there is this quantity that we can measure, that is, what is the width of this relation between different cells versus the height, okay? Now, if you have something that is actually planar, okay, that quantity, or if you have prisms, that quantity is going to be constant. But once that you have curvature, okay, that quantity, the aspect ratio between cells, is going to change, okay? Okay. So this is kind of the same that you have with these pieces that I distributed around, but in terms of the computer simulations. So these are the 4 cells that actually are having this relation. And as you can see with those shapes that you have in your hands, you will see that there is this kind of weird twist between cells that is actually the one that is able to achieve this neighbor interchange between them. So an ideal rendering of this kind of neighboring relation would be something like this. Okay. Now, to our surprise, and there were mathematicians involved in this kind of research, like Clara Grima, but that maybe you are familiar with her, or well, a number of them. To our surprise, this shape didn't have a name in math. So we have to come with a name. So this is the kind of the official reason that we actually we gave in the paper, but it's actually there is another reason that Luisma is involved in that reason. His last name is Escudero, so probably you will see why. But in any case, in the shape, as you will see, resembles something that appears in the thorax of beetles, like this one here. And this is actually the shape that we have to describe at the single cell level. That is kind of a prism, but with this kind of weird triangle over there. And we have to come with a name. The name that we actually gave to this shape was the scutoid. Okay. Now, something that you can actually see also in the numerical simulations is the following, that this is the longitudinal axis, this is what we call the transversal axis, and something that you can see for the cells, if you are actually looking the basal surface, so the one that is actually the outer of this cylinder, is that there is actually the appearance of these scutoids are linked to some geometrical descriptors or traits. Here, what I am plotting in this kind of histogram is this angle of the relations between different cells. And you can actually see that in the cells that underwent transition and scutoid in this talk is going to be the same. You will see why in a sec. This angle was small in the basal surface. So these kind of edges between different cells were mostly perpendicular. But in the cells that didn't underwent that transition or that didn't take that shape, they were kind of all over. Okay? So there is a geometrical information here that is important in all these things. So with this, we move to the lab to do experiments. And we chose a particular tissue that has kind of the same shape of a cylinder. Okay? And these are the salivary glands in Drosophila. That are these kind of these tubes, if you want, or these cylinders. Okay? This is just a cat, but it will be like a cylinder, as you will see in a sec. So these are these tubes that they can be segmented in 3D, so we can extract the 3D information of these guys. This is 4 particular cells that you can actually look in more detail. and you can actually peel off to see what is the neighboring relations. And actually something that we saw is that indeed these cells were packing following this shape that was predicted by the computer simulations. And when we also moved to the lab to see if these geometrical traits were also present in real tissues, we actually saw exactly the same. That when we account for this angle, for instance, here in the neighboring relations in that surface, the outer surface, also cells that have this shape actually prefer to align in one particular direction versus the other that were actually distributed all over. Okay. There is a lot of things going on here that I'm going to skip, but the question here, or one of the main questions in the paper, was why nature at some point decided to go with this particular shape and not with the prisms or the fruition that I told you before. What is the advantage for nature, for cells, to have this particular geometrical shape? And something that we realize is the following, is that imagine here, and I'm going to focus first on the experimental images. This is the relation between different cells, okay? And you can actually quantify here also the aspect ratio that I told you before. So how is the width versus the height for these cells? And you can see actually that as you move from one surface of the tissue to the other, it's actually changing, as I told you before. If there were prisms, normally this will be constant, okay? So in a schematic view, it will be like having relations between cells that will look like this. As I told you before, the width, the height, length of this small segment that I told you, we saw that there is a relation with it in the experiments and in the computer simulation, and the aspect ratio. So something that you can do is actually to account for how much energy you need in order to glue together, if you want, these cells. And you can account this by a simple calculation that will be, if you have a rope, or actually a series of ropes, that need to connect all the green points that you have there, okay? That is going to tell you more or less how much glue you would need to spend in order to connect these points together, okay? The length of these ropes is going to tell you what is the contact between the different cells, okay? This long expression here is actually very kind of easy to derive. It's just taking into account how that length changes as a function of the aspect ratio. So how much energy you need to spend in order to glue together all these cells as they change their aspect ratio due to the curvature of the tissue, okay? And something that actually can be seen as you move to the apical, to the basal part, from the inner part of that cylinder to the outer part of the cylinder, that is exactly the same as changing as the aspect ratio is changing, that when you are near to the apical surface, actually this configuration is the one that has the lower energy. But as you move to the other surface, there is actually this other configuration here is actually the one that takes less energy. So actually the reason why nature at some point decided to go with this shape when packing together cells when there is some curvature is just economy. If they have to pack together those cells, the amount of glue that they have to spend, if you want, the amount of energy, is actually less when you have these scutoids, okay? This is not just an idea that comes from a model, because short after we published this, there was a group that decided to do this experiment with foams. So in that case, they have these cylinders, they put foams together, and actually they saw that it was right. When you are actually accounting for the surface tension energy, it's actually minimal when you are actually packing these foams or these in this particular geometry, okay? And this is not something, again, that just applies to the particular tissue that we were looking, the salivary glands. And this particular example is actually something that I like a lot. So this is actually the Drosophila embryo. And in the Drosophila embryo, and this square here is the one that is kind of zoomed here, you can actually have part of the tissue that is pretty flat, the one that is actually marked in red. But there are other regions where there is actually some invagination, so where the curvature is actually very high. And you can actually see here that detail along that line in green, so that here the tissue is very flat, that here you have this invagination where there is curvature that is actually kind of dominating. Okay? You can see here more in details. And something that we saw is that actually in the part that is more flat, is actually the percentage of escutoids were like 15%, but in the part where there is actually curvature, the shape, 50% of the cells were actually taking the shape of escutoids. Okay? And you can see that detail about changing of those cells, the neighbors. So as you move from one surface to the other, how they changed or what became neighbor. Here is more clear, red and green, or the blue and yellow, blue and yellow. So exactly as I told you before with those, with that toy model. Okay. So, and we saw also in other organisms this. So we believe that it's a universal principle, if you want, in order to pack cells together, epithelia, due to curvature effect, that is actually linked to energetic constraints. So we published this in 2018. There were a lot of people involved. I'm going to mention something about them later. As I said, there are mathematicians, engineers, biologists. Luisma was actually a driving force in this project. And to our surprise, probably that is the biggest achievement that I'm going to have ever in my life, is that this went crazy in internet and in pop culture, something that I never would have suspected. I don't know if you know Steve Colbert. Colbert in the US is like Buenafuente here, if you want, or Wyoming, if you want. So it's actually a big part of the American TV culture. And actually, he mentioned...
So what is this hot new shape that's burning up the internet? Ladies and gentlemen, may I present the scutoid.
And there were other programs talking about this. Now, we are not getting rich with it at all. But now, if you go to internet, you can find almost everything with escutoids, like wedding rings, T-shirts, sculptures, paintings. So it's actually, we never imagined that it could go that far. Okay. Anyway, something that is interesting about escutoids is that if you look, and now I am looking on a single surface, on a plane, okay? Those cells from the top or from the bottom, it doesn't matter, is that you kind of have a puzzle with shapes or polygons that have different numbers. So there are cells with 6 sizes or 6 edges. Some of them have 7, some of them have 4, and these kind of things. Okay? And again, this is in 2D. Okay. Now, something that is actually pretty surprising is that if you go and check these tessellations in organisms, in epithelia, whatever organism, the histogram of shapes that you are going to get is conserved. So, different organisms are going to have kind of the same number of hexagons, the same number of pentagons, and these kind of things. Okay? That is surprising. Actually, there is a mathematical reason for this. that I'm not going to discuss, but if you are curious, we'll be able to tell you more. So something that you can actually, if you compute kind of the average of this histogram of this distribution, the number that you get is 6. And actually Euler knew that. There is an Euler formula that actually, if you apply it, Euler didn't know anything about these kind of things, but if you apply Euler formula, you know that you have to get 6. You don't know what is the histogram or the probability distribution, but you know that the average is going to be 6, applying Euler formula. In terms of the biology of the problem, this is telling you that in the plane, those cells on average are talking around with 6 other cells. So the 2D connectivity of those cells is actually 6. Now, if cells were, as I said before, presence or frustrum, when you look this, what is the average connectivity in 3D, it would be 6 also, because they are not going to change neighbor in all the depth of the cells. But actually, now I told you that the cells, they are not going to look like this. When you have scutoids, the 3D connectivity is going to be different, because I mean a simple example here is that the red cell has a connectivity here of 2, okay? It's connected directly with the blue and the yellow, but in this other surface, because of the scutoids, it's also connected with the green. So the 3D connectivity, the red cell, is actually in this case connected to the 3 cells, okay? So scutoids, when you look in 3D, are going to change the amount of people or the amount of cells these cells are actually talking to. It's not going to be, cannot be 6 anymore, okay? Not only that, it's a quantity, the connectivity or the number of 3D neighbors, that is going to change that as you move through the coordinate, the apicobasal coordinate, okay, through the radius. As you move through the radius, you are going to start connecting with more cells. So it's something that depends on this coordinate, the apicobasal coordinate, and we know for sure that it's going to be equal or larger if you have scutoids than 6. Another question is that how can we figure out what is the number of or the 3D connectivity of these cells in 3D? And you may ask, why is this actually important? Well, it's actually biologically very important to know what is the connectivity, because when you have juxtagram signaling between neighboring cells, that really depends on how many cells you are talking to. So the intensity, if you want, of the juxtagram signaling, how cells are actually communicating with each other, obviously depend on how many cells you are actually talking to. And this is actually a problem that has been highlighted very recently in a number of cells. So this is a paper published in Science in 2020, and actually talk about the contact area dependent cell communication. In this case also because they were able actually to check out the 3D geometry. Sorry. Moreover, now there is a lot of interest in these phase transitions, that actually how those mechanisms can be important in embryonic development. And in this recent paper in Cell is that actually they saw that the kind of the viscosity of the tissue, how rigid a tissue is or is not, has actually a dependence with the connectivity of the cells. So actually knowing the connectivity is telling you a lot of things from the signaling viewpoint, but also from how the shapes, again, can be regulated on a 3D environment. So something that we check out is that with these Voronoi 3D models, what we have is these computer simulations that are rendering you the 3D structure of those cells. Remember, we have this apical radius RA, the basal radius RB, any intermediate value is going to be R, and something that S, the surface ratio, is this dimensionless value of the radius, okay? And something that we can check with these computer simulations is how, as a function of these surface ratios, so as a function of the apical basal coordinate from bottom to top, okay? How what is the average number of these apicobasal intercalations per cells? Apicobasal intercalation is nothing that this neighborhood change that you have between different cells due to the scutoidal shape. The scutoidal shape actually facilitates that you exchange neighbors. So here we are actually accounting how many of those intercalations between cells in a 3D environment happens as a function of this coordinate here, okay? And this other scale that is in the vertical scale is the different Voronoi tessellations that you can actually have, the different models that you can imagine that you can actually have in these computer simulations. The important thing here is that when you have big one, that means that the cells, or there is going to be a lot of disorder, meaning that this distribution of neighbors on the plane is going to be broad, okay? And as you increase this quantity, you are getting more and more ordered cells in 2D. That means that the histograms, the polygonal histogram in a surface is going to be more and more ordered and more peak, around 6, that I told you before. But something that you can see here is that it doesn't matter that much what is the order that you have in one of the surface, that the apicobasal intercalation seems to not care really about that, okay? Maybe when you have a very purely random situation like in this one, but in the rest is more or less constant. It doesn't depend, the number of 3D neighbors doesn't depend much on that, okay? No, I don't want to do that. I'm going to go back. Something that you can actually also compute in these cells is actually energy, okay? And we can actually extract that from the geometrical properties of the cells. So this will be like elastic energy, but in 3D, okay? So it's like how if you have a spring, but in this case it's a volume that you are actually deformating, and you have a particular ideal or rest shape of your spring, that in this case is a target volume of the cell, but then you have that there is deformations because you have these cells that has to bend in this geometry, so there is a particular elastic energy accumulated in the cells. You can also accumulate energy in the forms of the lateral cell area that is going to tell you something about the surface tension energy, okay? How much area do you have in those cells? And there is another important quantity that you can actually check out, that is the so-called related with the apical cell perimeter, that is related with this polarization that I told you about cell, that the mechanical interactions are morely on the apical sides, and in particular there is one contractile ring, okay, that has a lot of energy, so it's like an spring, but in this case, like if you have a, what is the word in English? Goma. I don't remember how to say. Rubber band, okay? Like if you have a rubber band in one of the surface of those cells. So on top of knowing or characterizing as a function of this coordinate how many intercalations you have, you can actually account of how much energy or how the energy change as a function of this coordinate too. Something that we realize is that surprisingly, the cell area doesn't depend much on the disorder that you have in those cells. This Voronoi scale that is here, the Voronoi class. The cellular volume fluctuations that are actually related with elastic energy does, okay? The more disorder, the more accumulated elastic energy you have in those cells, okay? And the apical cell perimeter on average is more or less constant, okay? So now something that we did in order to understand how cell connectivity is connected with these intercalations and these energetic terms is doing cross correlations. So cross correlations, if you are not familiar with it, is like if you have two signals, okay, and you displace one signal versus the other, and you find what is the displacement that is giving you more correlation, okay, between them. So something that you can do is that, okay, we have a signal that is the amount of intercalations that we have. We have other signals that we have the amount of energy that we have. And now we try to find if there is a correlation between them. Okay? This is what you have here. The lettering is not the best. But basically what we are seeing here is that the correlation between intercalation and elastic and energy terms is actually maximal for the surface tension. And this totally agrees with what I told you before, that the amount of glue that you have to spend. Okay? So maximum correlation between intercalations and energy is due to the surface tension terms, but elastic energy, that is the red curve, also plays a huge role. But another thing that we actually accounted is not only the cross correlation between them, but the gradient of them. And I'm going to explain you this. Is here you are looking about the derivative of the intercalation, so how they change as you move as a function of the radial coordinate, and also how the energy changes as you move as a function of the radial coordinate. So we are looking about the cross correlation between the gradients, the derivatives in this case. And actually what we saw is that, again, the maximum correlation is achieved for changes in the surface tension energy, but also there is a lot of change when you consider changes in the elastic energy. But the important thing here is that here you see that this signal is peak in 0. That means that the two signals are actually the maximum agreement between them is when the displacement is null between them. Okay? But when you look at the cross correlation with the derivatives, you see that the cross correlation, maximal cross correlation, is achieved for not null displacement, but for a displacement of one signal versus the other. The important thing here is that with cross correlation you can actually suggest if there is or if there isn't a reason, or actually what is driving what. It is actually intercalations that are driving changes in the energy, or are changes in the energy that are driving the intercalations. That is the advantage that you can actually look with cross correlation. This information here is actually that changes in the energy, because there is this positive lag, are the ones that are actually generating changes in the intercalations. Okay? We can actually connect all these ideas also with this topological information about cells. So something that you can actually plot is actually, what is the polygonal class, so how many cells with 4 edges, 5, 6 you have in a surface, with respect to how many neighbors or the polygonal class of the same cell in the other surface of these tissues. Okay? If all the cells were prisms, like the red ones here, all the histogram will be accumulated in the diagonal, because cell will have the same number of neighbors in one surface and in the other. As soon as you have these scutoidal shapes, you are going to spread this histogram in this direction, in the cross or counter diagonal. Okay? And you can actually measure that spreading with something that is called the spreading coefficient, but basically it's telling you how much it is deviate of being purely in the diagonal. And this is what we call the spreading coefficient. So the spreading coefficient is actually also a measure about how many scutoids you have in the system. And we can actually plot also the spreading as a function of this radial coordinate and as a function of the disorder. And actually we saw that there are changes here also depending on the disorder of the surface, how they accumulate the scutoids. And we can actually also perform the cross correlation analysis in order to understand how many scutoids you have in the system as a function of different energy terms. And we saw that in this case also there is a huge or larger correlation due to the surface tension. Okay? So everything check out or is in agreement with this idea that I told you before about the amount of glue. But also elastic energy, we saw that it's playing a role. When we compare these experiments, this is in vivo. This is kind of the 3 histogram where you can actually measure the spreading, the cross correlations between energy terms, the cross correlation between actually these gradients that I told you before. This is the results that we got. We compare this with computer simulations. There is one particular disorder and one particular side of those computer simulations that is given the best agreement with experimental results. And actually we saw that the behavior that we got is actually very similar. Okay? So we have a very good computational model reproducing the experiments in that regard. In terms of energies, disorder, the spreading coefficient that I told you before, and so on and so forth. Okay? And something that we saw is one particular or interesting thing about how you accumulate neighbors or how you accumulate connectivity. That is this principle that we call the purge get richer. So let me explain a little bit more this. This is as a function of the polygonal class that those cells have in one surface, in the inner surface, how many neighbors you accumulate in total, okay? The 3D neighbors that you win, depending on the number of neighbors that you have on a surface, if you want. And something that we saw is the smaller number of neighbors you have on a surface, the more neighbors you accumulate. The net gain is larger. So if you have 4 neighbors in one surface, you accumulate way more than you have 8 neighbors on a surface. Okay? That is why we call the poor get richer. So that is telling you that the number of neighbors somehow has to be connected also with this energy cost in order to win or not win a particular neighbor. So for doing that, we develop another model, and let me pull this in a different context of the previous model, because the previous model gave us an explanation about why nature decided to go with scutoids. Now we want to do a model that can be quantitative in order to tell you how many neighbors you are going to win in a surface depending on the energy that you spend. So it's actually quantitative in that regard in terms of what is going to be your connectivity on 3D. So this model is based on a stochastic model that takes into account like a Markov chain, okay? That is telling you that if you have 4 neighbors, you can stay with 4 neighbors, or you accumulate 1 neighbor more, and so on and so forth. Something that we account in the model is this principle that I told you, the poor get richer. So if you have 4 neighbors, the amount of the energy barrier that you need to jump in order to win 1 neighbor is going to be less than if you have 6 neighbors. So the height of that barrier is going to be larger. Okay? Not really important, or well, actually it's really important, but I'm not going to enter into details how we did that. But actually what I'm going to tell you are the results. This is the in silico model. The colors are telling you the histogram, but in this case on 3D, of number of 3D neighbors that you have as a function of the apicobasal coordinate, the radius if you want. The red dots are actually the average number of 3D neighbors that you have. This is the prediction of the model, okay? Computer simulations model. And in this histogram you have the error, okay, that is scaled from 0 to 100%, 0 to 1. And this is in vivo, what we saw. And this is the in vivo measurements. This is the biophysical model. As you see, there is a perfect agreement between both the in silico and the in vivo simulation with the model. And the error is actually very small. So how can we actually test not only we have a model, but we can actually challenge the model? something that we did is, remember that you have this addition that we saw that is actually kind of the main driver of these intercalations. So something that we can actually do is try to remove as much as possible this addition between cells. This is actually removing the addition by actually removing a particular addition molecule that is called icahedrine. So we did that. So we repeat the experiments. In this case, this will be the profile as a function of from top to bottom or from apical to basal, the amount of cadherin that you have in the wild type or the amount of cadherin that you have in these cells that are actually mutants for this addition molecule. And actually what we saw is that reducing addition actually leads to a larger concentration of intercalations as a function of this coordinate. Okay, so that means that if you remove the glue, you actually facilitate this exchange between different neighbors. What actually makes sense. Now, in the context of the model that I told you, that would mean that if this is actually true, the amount of energy that you have to spend has to be lower. Okay? So we actually check. This is in vivo. This is the model. This is the agreement or the error that we have in the model. This is the amount or the height or the energy barrier that you need to jump with the wild type. The red curve is the amount of the height of the barrier that you actually have to jump in the mutant, that is slower. And this is the amount of the barrier that you have to jump in the computer model. And I can tell you why later if you ask me why actually you don't have such math energy. But the important thing here is that when we look into in vivo results, our model actually confirms that the amount of energy or the barrier that you have to jump is actually lower when you decrease the addition. There is a particular approximation to this particular model that I told you before, that you can actually solve exactly the number of 3D neighbors that you have as a function of the apical basal coordinate. In that case, you have to assume that the energy barrier remains more or less constant, something that we saw is not the case in general in vivo. And in that case, what we saw is that the number of how you accumulate neighbors in 3D follows a logistic behavior. We call that rule the Flintstone law, but I think that we are not going to use that anymore in the paper. So all this second part is actually right now under review in cell system. If you are curious or more curious about it, we published a review last year in development about trying to give you an historical overview about how people have been interested in understanding the 3D shape of epithelial cells or cells in general when they pack together, forming organisms. The take-home message would be that escutoids made possible the minimization of the tissue energy and actually create a stable way of pack cells together. That we believe that are nature's solution to achieve epithelial bending in an efficient way. That we have found a novel quantitative principle that explains how cell connectivity actually can be achieved or quantified during morphogenesis. And we think, honestly, that all these results are going to be important when you try to understand better the formation of organs or even in organoids, artificial organs and these kind of things. There is a lot of people that actually deserve really the credit, not me. A lot of people involved in this work, as I told you before, mathematicians, engineers, biologists, you name it. But there is 4 persons that I would like to thank in particular. So one of them is Pedro, the other one is Pablo, and the other one is Samira, and obviously the other one is Luisma. Luisma is the other PI that has been involved in all these works. Pedro is, so these were PhD students. Pedro is now a postdoc in Cambridge since very recently. Pablo is a postdoc in UCL in London, and Samir is a postdoc in Johns Hopkins. And Luisma and I are here. So Luisma is in Seville, I am in Valencia, so if you want anything from us. If you want to follow me in Twitter, or if you want my email, this is the one, and the funding sources, NSF, NIH, ministry, and also CSIC, and that is how I want to finish. So last year, you know that there were these white papers or books that were published by CSIC. I was involved in this particular one in volume 2, Origins, Coevolution, Diversity, and Synthesis of Life. There is a meeting here next month about this kind of issues, because due to this particular white papers or books, network has been organized. It is called Life Hub. If you want to find out more, you have to check that out. We are open. This is a huge network of scientists and researchers. If you are interested in all these kind of things from different viewpoints, many different viewpoints, if you are trying to find collaborations. So if you are interested, just check it out, and we will be very pleased to have you there. And that's it. Thank you very much.
Okay, thank you very much, Javier, for this excellent talk. And congratulations for this new concept of escutoid that is so famous. So now, now is time for questions, and those people attending the seminar, excuse me, by YouTube, you can write your questions down in the chat, and we will ask this question to Javier. Now, please, if you. Pablo. Oh. Sorry.
Hi, Javi. Thank you very much for your talk. I have two questions. One is, please, could you comment more about the experiments, the microfluidic experiment, just for the students, in order to get an idea how do you get the experimental values with the experiments. And the second one is, correct me if I'm wrong, but your studies are in a single layer. Is that right?
It's a monolayer of cells.
Okay, so my question is, what happens if you add two? Because probably this is going to have important implications in terms of elastic properties and so on. And probably there also the scutoids have an important role there.
So I'm going to repeat the question, because so there was like kind of two questions. So one question is how to we can actually get the data somehow. The other question is that I have been talking about epithelial monolayers, and the question is, well, what happens when you don't have just a monolayer, but several layers of epithelia, right, or epithelial cells? So as for the first one, I have to confess that I did nothing. Okay, so that is the huge patient work from Luisma's group, getting all these images that are actually stacks that are obtained from microscopy, and from that, how do you actually reconstruct the shape of the cells involve many different things. So at first, you need very good markers in order to distinguish what is a cell, what is an individual cell with respect to another one. So to distinguish individual cells. So the markers are important. The other thing is the imaging. So how you actually image this. And of course, in the microscope, what you do is you take slices if you want. So imagine that you have a confocal. But then from those images, you have to go and reconstruct a structure that is in 3D. And so in this, there is a images processing pipeline that has been a lot of work. The last version that has been used, that is in the paper that hopefully we are going to publish soon, involves also machine learning. So how can you actually reconstruct in a very efficient way, this kind of thing. Now, once that you have the shape of the cells, then you can measure everything. Like it is, I'm not going to say that it is easy, but you can actually say how many area they have, how many volume they have, what is the surface that they have, what is the polygonal shape that they have. But the bottleneck here is how you go from the experiments to the quantitative data. That is an amazing amount of work. Yeah. But all the details can be, Luisma will be actually the person to respond to this. I'm sorry, my response is taking too long. The second part, that is an incredibly interesting problem, but I don't have a clear response to that. A lot of things can go on. And there is some people that is actually working on that issue, like different stacks of epithelial cells and how the shape are changing and these kind of things. But I can tell you more. Yeah.
Okay, more questions. Yeah, Miguel.
Thank you very much. I have two questions. One is, in this situation, this kind of structures are also present in other kingdoms like plants or fungi. And the other one is related with the shape of the cells. I was thinking about, for example, fibroblasts, that which the dimension of the width of the cells is so. It's the contrary situation that the epithelial, which are high. This dimension is the higher one, and the other one is the other. You have a minimal size when you have this situation to obtain these structures.
So, and correct me if I am wrong, there are two questions like, so there is this shape appearing in plants or and the other question was like, how important is the height of the cells? Yeah. Okay.
Well, and the other one, the kingdoms, the different kingdoms have the same.
Yeah. Excellent questions, both of them. We believe, and well, that is a belief. It's not something that we can prove, but we believe that. If you think about it, it's like what I told you so far or in the seminar is that with these shapes, you can achieve more efficiently bending, shaping things, curvature, this kind of. So we believe that there is an evolutionary reason for these shapes to be present. When we look in plants, we haven't seen any scutoids. For instance, does that tell you that from that viewpoint, plants, I mean, it's a different story, right? So they have, in plants, cell walls, the rigidity is actually way larger. So actually, it's like they are stuck. They cannot actually achieve more shaping creation, if you want. So they are limited in that regard. We haven't looked everywhere. What we know is that in epithelia, in higher organisms, as far as we know, it's always there. Plants, we know that they're not. I think that fungi, they do. So I think, and we believe, as I said before, that there is an evolutionary reason for all of this. This is an excellent question. We do not know what are the specifically the gene pathways or sequences that are actually connected with that shape. It would be extremely interesting actually to be able to identify at the genetic level what is the mechanisms, and then look in the tree of life, if you want, try to see if there is a divergence in that regard. Okay. The other question is yes. So we are able, we have been seeing these shapes in particular in the salivary glands, because salivary glands are actually very tall. If you look to cuboidal epithelial cells, probably you wouldn't see much. So you really need this apicobasal coordinate to be long enough in order to give the opportunity, or actually that the energy that you need to spend, if you want the change in the aspect ratio that I mentioned before, has to be long enough in order to make economically energetic, viable, to change the shape. Yeah.
Very nice talk, Javier. And what about disease? I mean, would you be able to relate a not healthy tissue with some of these parameters? Have you looked at it? Are you working on that or something?
What I do know is because, and Luisma was working into that, from the point of view of surface, so not on 3D. So, well, let me repeat the question. The question was like, can you connect these shapes or not with disease somehow? So what you know is that when you look at the cellular organization between cells in a surface, in a plane, how many pentagons you have, how many hexagons you have. Some pathologies have been linked with how that histogram of neighbors actually changes. So there is clearly in that regard a relationship between pathology or a disease and cellular organization. Also, I told you that the mutant case, when you are actually decreasing cadherin, in that case we saw that the number of accumulation of 3D neighbors can actually be different and these kind of things. So clearly when there is a particular mutation or problem in the mechanical properties of cells, you are going to change the organization of them, you are going to change the 3D neighbors that they have. So more likely there is a relation, but so far we don't know any particular disease that is telling you that less scutoids or more scutoids are giving you to be more susceptible to this or that disease or whatever. We don't know. But probably there is. But we don't know.
Yes, and related to this question, Douglas Lawrence makes a question. Thank you very much for this most interesting talk. I would like to ask you, cancer frequently impacts epithelial tissues. Do you have some insights into this? Cancer growth alters the packing geometry.
Well, thanks for the question. As I was just responding, what it is actually known is that the cellular organization is, as he pointed out, totally in relation with how the cells organize together. But in the specific context of the 3D organization or scutoids, on how that is related with cancer or not, no clue. Very interesting question, but I don't know.
Thank you. Any other question? I have a question, a comment. As far as I know, epithelia are very dynamic tissues in terms of cell division, cell renewal. New cells appears and the old cells die. How this the geometric aspect of the epithelia combines with this dynamic aspect of physiological dynamic aspects of epithelia.
Okay. I am glad you are asking this question. Thank you very much, because actually that is normally the question that I do expect to get from the audience. Like, everything seems like pretty static in all these things. Actually, it is. In all the things that I told you, everything is static. We were able to work with salivary glands because in that particular tissue, all divisions are stop at that, so everything is pretty static. But as you said, epithelia are actually pretty dynamic, and they can actually change. There are cell divisions. There can be migration. So how this fit together? We are actually working on that. And so one of the students that actually just recently joined the lab is trying to develop computational models actually that not simply reproduce an static situation or packing organization of tissues statically, but also dynamically in 3D. There are not so many or computer simulation tools to simulate 3D tissues, epithelia. Actually, most of them put all the scutoids under the carpet because they are difficult to deal with in computer simulations. So many people disregard those, like, well, we don't care that much about those. We do care, and we are actually working. So there is going to be a lot of probably transient scutoids. That has been known for a while, that you can have these apical-basal intercalations that appear and then disappear. So you go from these scutoids to a prism again, or from a prism to an scutoid and back. So probably that is going to happen. Some of them are going to get stuck. That is why we are trying to develop these tools also to address better the dynamics of the tissues. Yeah.
Because I'm not an expert on this, but I think this is quite relevant, for instance, in colorectal cancer, because the intestinal epithelium of colon is continuously challenged by external factors.
Yeah. The renewal is very rapid, actually.
Exactly.
Yeah. Absolutely. And actually what happens is that in that case, curvature is actually critical also, because you have all these crypts.
Exactly.
So actually probably the epithelial organization there is going to contain scutoids, but it's actually changing dynamically very rapidly. So yes, very interesting problem.
Okay.
Yeah.
Thank you very much. So if there is no more questions, just to finish this up, this is a small present for you.
Well, thank you. For my birthday.
Yeah, it's something for your birthday. Yes, we knew that it was
Okay.
No,
Okay.
it's a joke. This is a present for you as a speaker, as a participant in this.
Okay, thank you very much.
Okay, thank you, Javier.
Thank you. Me alarga un montón, ¿no?
La historia. No, está bien. Te contamos la historia porque esto también está relacionado.